All right, so here we are for Chapter 8, Part B. We're going to look at HIV in this section, and then in Part C, we're going to look at some other infectious diseases. So to look at HIV, we do want to look at a history of it, and you'll get a little bit of this out of your podcast, but HIV was originally found, we've been able to trace it back to non-human primates. It was at that point called SIV, and that was in Africa. And we think that at that point the virus mutated and perhaps was transferred to a human through a cut in the blood or consuming an organism. So we're not 100% sure, but HIV was found in non-human primates and then made its way into humans after being mutated. The exact dates of the first human case are still being investigated. People are still looking into it. We're discovering more and more each year. So it's around the early 1900s, we think. At that point, there wasn't a ton of huge travel. I mean, travel was increasing. People were living more in cities. People were living in larger communities, maybe being a little more promiscuous. But then there was also international travel, and people were able to move a lot faster through the world. So the first documented death of HIV in the United States was 1969. And then HIV continued to increase from there. People became more aware of it. And it was discovered, it took us a long time to figure this out, but HIV was found to be the cause of AIDS in the 1983-1984 era. It took some research putting together there. So once we knew that HIV was causing AIDS, then we were able to look at the HIV to try to prevent it or try to treat it and try to move forward that way. Right now, approximately 34 million people are living with HIV today, and over 36 million people have died since the beginning of the epidemic. So remember, it's an epidemic because it's more than we would have anticipated in a certain area, which happens to be quite large, in a certain amount of time. Our map in the bottom left tells us where the HIV proportions, where the adult population, the percentage of that population that has HIV. So we can see it's concentrated in Africa and the Americas, and then a little bit less in Europe and Northern Asia, and then sort of the Middle East, and then finally Southeast Asia a little bit. So there are 3 stages of HIV infection. There is category A, category B, and category C. Those could also be called stages instead of categories. So what's going on here is that each of these are marked by a certain thing. And note that this years since infection, this is not perfect. It's not at exactly 3 and exactly 7.5 for everybody. We do see similar progressions, but those numbers don't have to be exact. And what we're looking at here is a graph with 2 y-axes. And so the blue line, we want to read the numbers on the left, which is CD4 T lymphocytes in blood. Now, we didn't go into what CD4s are, but they are a type of T lymphocyte, and that should be familiar to you. So CD4 cells, you can think of them as a T cell. So here's my CD4 cell, which is also a T cell. And it's a white blood cell, it's floating around. This is the cell that HIV infects. It doesn't infect your B cells, it doesn't infect your monocytes, it doesn't touch your red blood cells, it doesn't touch your liver cells, it doesn't touch your epithelial cells. It doesn't do anything except invade and ultimately debilitate your CD4 T lymphocytes. So that's why our stages of HIV infection are very closely linked to the amount of CD4 T lymphocytes that you have, shown in this blue line. Now, the other line that we're looking at here in our graph is the red line, and that is the amount, purple line. That's the amount of HIV in your plasma. How many actual little viruses do you have in your plasma. So that's what we're looking for in this purplish line. So you might notice if you just like take a step back and you mostly ignore the very beginning, but just kind of take a step back, you might notice that the blue line and the purple line are mirrors of each other. If we were to draw a line along the middle, right, we see when one is increasing, the other is decreasing, and vice versa. So that's what we want to pay attention to. So you get infected with HIV, well, hopefully not you, but somebody, then that it replicates and it replicates and it replicates and it replicates, and while it's replicating, it's infecting these cells right here. So the number of CD4 cells that you have starts to go down. And now the body is able to fight the infection a little bit, so the CD4 cells are still suffering. They're still going down because they're fighting this off. But HIV is being killed, and this line is going down. But unfortunately, once the HIV has established itself, the body isn't able to completely fight it off, or else this would go all the way down to 0. So what HIV is here, what's left here, is able to slowly replicate. And as it replicates and divides and spreads, the number of CD4 lymphocytes you have that are trying to fight it, but they can't, are going to go down. Okay? So when you get to a point where your CD4 lymphocytes, that count in your blood is below 500, that's when you've reached the chronic phase. Before this, you were at the acute phase, meaning right at the beginning. Up until this point, you're at the acute phase, but once that CD4 count gets down to 500, you're at the chronic phase. And then we're going to just see this continuing. So the more HIV that you have, the less CD4 you'll have, and when those CD4, again, when they get to 200 or below, that's when you've entered category C, and that's what we actually call AIDS. This is when you officially have AIDS. Up until this point, you've had an HIV infection, and in category C, it's progressed to having AIDS. Again, these numbers in years could be flexible. It would depend on the person. We do have some treatments that are available now that extend this for a much longer amount of time. But this is sort of just a generic what it might look like, or what it would look like without treatment. So that's what we've got for the stages. And so we finally learned these things. And then, well, and at the same time, people were trying to say, okay, how do we stop this? We know this is killing people. We've got to understand the HIV life cycle if we're going to have any hope of actually helping people and helping prevent the spread of the disease, helping prevent the progression of the disease, perhaps even reversing it and killing it off entirely. We have to understand its life cycle. And so that's what we have here. And there are a number of steps. They are listed on the next slide in text if you want to look at those at the same time. And there is an activity in your assignment where you're reorganizing the steps just to practice using this information. So it starts with attachment. Now, HIV, the V in HIV stands for virus. So virus tells us we've got some sort of DNA or RNA, and we've got a capsid, and we might have some spikes. And we can see all of that in our image, right? We've got DNA and RNA, we have a capsid, and we have some spikes. Okay, so those spikes are going to attach to your CD4 T lymphocytes, which is what we see in the big picture here. So we attach. The second step is fusion. So this is essentially, it's a version of endocytosis and exocytosis, right? So we can see that the capsid of the virus has joined with the plasma membrane of the host cell, and we're technically spitting out this green capsid here. So your envelope has formed with the plasma membrane. We're spitting out the capsid. So we have fused the virus to the plasma membrane. So step 3 is entry. Okay, in step 3, we see that the capsid has broken down, and we see that in this case, in HIV, we have viral RNA. The virus has RNA, not DNA. And as a result of that, we have it exposed, floating around in the cell. Now, let me pose you a question. Do you have RNA or DNA in your nucleus? You have DNA. Awesome. So if this viral RNA just tried to wiggle its way into the DNA, it would be kicked out. Our body cells would recognize it as being foreign. It would only be single-stranded instead of double-stranded, and it would never be able to fit. It would never be able to work. So the only way for the HIV RNA to get into our DNA is something called reverse transcription. Now, if you've ever taken other biology classes, you might recognize the word transcription. Translation and transcription was turning DNA into RNA. This is just the opposite. So we call it reverse transcription, is turning RNA into DNA. And you might be wondering, why do our cells do this? And it's actually not our cells that do this. This little tiny blue dot here on the viral RNA, that is the enzyme that's going to do this. And that enzyme was previously inside the capsid of the virus. So the virus actually makes its own reverse transcriptase enzyme that just hangs out with it until it gets into a host cell. So the viral enzyme reverse transcribes the viral RNA into DNA, which we see in orange, and that DNA is going to enter the nucleus through a nuclear pore, and we're going to go through step 5, which is integration. That viral RNA integrates itself into our DNA because it now is technically DNA. It's been reverse transcribed into DNA, and it's in our DNA. And your cells don't even know it. That's the thing. And that's kind of, I guess a little scary, but I mean, it's cool on the side of the virus, but from our perspective, it's not so good, right? So the fact that it's actually just going to hang out in your cells at this point, and your cells aren't going to notice. There's just going to be like, oh, that's part of the DNA, right? It's not like it's actually a different color, or even if it was, your cells aren't able to, recognize different colors. So your body just has no way at this point of even knowing that this isn't its own DNA. So what happens at this point is step 6, biosynthesis and cleavage. Now that sounds really complicated, but biosynthesis just means we're making biological things. We're making proteins, and we're making the capsid, and we're making more viral RNA. Those things are all those organic macromolecules. We're just going to make a bunch of those. So that's what we see in actually a lot of this image. We see the viral RNA being produced and coming out over here. We see other parts of this viral DNA other parts of the viral RNA is being transcribed, not the reverse transcribed, but translated then into a protein, which is probably going to end up being, this little blue dot over here. And then we see the capsid being made. So all of these pieces, your body cells technically made. It made it based off of that chunk of DNA from the virus, but your body doesn't know that, so it just keeps making those pieces. And then those pieces come together in assembly, and they make a capsid. You've got your viral RNA in here. You've got your proteins, your enzymes, your capsid. And then at the end we have budding, where the capsid comes out. It has spikes on the outside of it because your body made spikes too. Your body's membrane made those spikes. It was one of these things came up. That's what was actually formed with this spike. So now it has its own spikes and it buds. This is just a form of just budding off of the membrane. It takes your own body's membrane with it. And now over here we have an HIV virus. It has everything that it needs to infect another cell, and it's just going to keep going on its way. So here is what we just said, but in text. HIV life cycle, there is attachment. The spike binds to the cell's receptors. Fusion. HIV fuses with the cell membrane. Entry. Uncoating occurs. We uncoat the capsid, and that releases the RNA. Reverse transcription occurs, and that RNA is reverse transcribed into DNA. Integration is when that new DNA becomes part of the host cell's DNA in the nucleus, and that's when we call it a provirus. Biosynthesis and cleavage is next, when the host cell produces more viral RNA and viral proteins. Then assembly occurs when all of those new RNA and protein and new parts are assembled into viral particles. And then budding happens, where the viral particles bud from the cell membrane. And finally, we have transmission, where that virus is passed to a new host. So you might be, that's basically what the HIV life cycle is. And we're able to know that now because of lots of hard work that went in. And we can use that to try to learn about how to prevent it and how to test for it and how to then cure or treat it. So transmission of HIV, many of you have heard this before, I don't expect I'm going to be shocking anyone here, is through one of 4 big methods. Sexual contact, dirty needles, or previously used needles, a blood transfusion, or mother to baby. Now keep in mind, a blood transfusion could of course be donated blood being given to somebody. In the United States, we absolutely test for that, and we do not allow any blood with HIV to be donated or used. But that blood transfusion could be something very casual, like somebody accidentally having a cut on their hand and interacting with someone else's cut, and that blood gets transferred to someone else. So any of those 4 things is a way that HIV can be transmitted. And it's really important to know that HIV is not passed through casual contact. Someone who has HIV, you can absolutely shake their hand, you can give them a hug, you can even give them a kiss on the cheek. HIV really doesn't occur in saliva unless that saliva, unless there's cuts in the mouth making the saliva bloody. So you could even kiss somebody who has HIV and not have that transmitted to you. So all of those casual contact things are things that make people with HIV infections feel human and feel like they're still part of the community. And so your risk of infection is none with those casual contact sorts of things like hugs and handshakes. Prevention, of course, is going to be not doing these things we mentioned. So abstinence, or having sex with one uninfected partner, or using proper and consistent use of condoms to break down on the sexual contact side of things. Of course, with dirty needles, prevention would be not using those. For a blood transfusion, the prevention of that would be don't touch anyone else's blood ever, which is a good rule of thumb for everything, because there are many blood-borne diseases, not just HIV. So don't touch anyone else's blood, only get a blood transfusion if you can help it, in a place with obviously safe methods and they've tested the blood and all of that, which in the United States you can pretty much rely on that, unless you're somewhere super shady. And then testing, from mother to baby, that transmission, we have now been able to create some new treatments to try to really reduce that happens. So that's our best prevention, is to treat the mother while she's pregnant to really try to prevent that transmission to the baby. Testing then for HIV is how we test for antibodies. So antibodies are going to form about 2 weeks to 6 months after the first exposure. And part of the challenge with this is that for some folks it doesn't show up until 6 months after exposure. So someone might be concerned that they were, maybe there was a night and they didn't use a condom, and so someone's worried they maybe got an STD. So they go to the clinic a couple weeks later, or even a couple of days later, to test for it, and it comes back negative. And that's great, but that person should absolutely go back 6 months later and get tested again, because it's possible that the antibodies just hadn't formed yet in response to it, and so you might be infected but be unaware of that. So absolutely, if anyone you know is ever worried about potentially contracting an STD, which would include HIV, make sure that you recommend that they do get tested right away, but that they also go back 6 months later and get tested again, just to be certain. So HIV treatment, what do we have to treat this, this disease? We have created, we've been able, now that we know the life cycle, to create drug therapy. So essentially we use a combination of drugs to try to inhibit HIV replication. And the idea is that we're trying to, if the HIV can't replicate, then it can never get to the budding stage, and if it can never get to the budding stage, then it can never transmit to anyone else. And that also includes it can't transmit to other cells that you have. So we're trying to essentially break it down. So if you look back at those 9 steps of the HIV life cycle, these drugs are trying to stop all those different stages. We're trying to prevent attachment. We're trying to prevent reverse transcriptase. We're trying to prevent entry. We're trying to prevent the biosynthesis and the fusing with the DNA. We're trying, these drugs, it's a combination of them, and they're all trying to prevent different stages. In the hopes that with all of these different barriers, it would be very unlikely for an HIV to make it through all of them. And those treatments have been fairly successful at this point. There are people who live very long, healthy, otherwise healthy lives with HIV infections. The other benefit of this is that they can be used to protect someone who might be in a relationship with someone long term, but one partner is HIV infected. And the HIV infected person can take these and still have a relationship with that person, but also while protecting their partner. The other thing that we have looked at doing, of course, is creating a vaccine for HIV. Scientists have studied over 50 preventative vaccines and 30 therapeutic vaccines, and so far none of them have been successful. And there's a few challenges in making an HIV vaccine that are really causing issues for scientists trying to do this. One of those is that no vaccine is 100% effective at preventing entry into cells. So that in itself is a challenge. The other thing is that HIV viruses are genetically different locally and globally. So even if you did get a vaccine for one area, that would only give you short-term protection, because what we'd need to do to make this global is make a vaccine for every form of virus there is, every genetically unique version. We would then need to get it everywhere around the world at the same time, and within a couple of days every single person would need to take the vaccine. So obviously that's a tall order, and we would need some serious global cooperation to make that happen. Another challenge is that HIV, obviously, you guys saw this, inserts itself into cells. It's inside the nucleus, and it hides from your immune system. So it's really hard for your immune system to find it, and as a result, it actually makes the vaccine a little trickier too. And we don't have an ideal animal model for looking at the vaccines. And just like for most people, it would be considered unethical to take people with HIV infection or to even give them the infection. It would be unethical to give someone a vaccine, a trial vaccine that says, hey, you're protected from HIV, go have a lot of fun, go do whatever you want, and then that person thinks they're protected and they go out and maybe they're not, and now they've contracted this disease. So that's not possible with humans, and a lot of people would consider that unethical with other animal models, and so we're not able to use that method.